Alrighty, my name is Hashem, and today we'll be speaking about the utilization of a CRISPR-Cas9 system to produce a mutation on the myostatin gene in Korean beef cows. So why was the myostatin gene specifically targeted? So myostatin is a gene that codes for the growth factor protein myostatin. It's a negative regulator of muscle growth, and when active, leads to less skeletal muscle differentiation. This gene is found in all mammals, and mutations in this gene are observed as well in the case of the Belgian blue cow and Piedmontese, which have an ineffective copy of this gene, leading to increased skeletal muscle differentiation. So why is that helpful? Why are researchers targeting this? So as the global population increases and demand for meat increases, there becomes an increased need for trait-improved cattle. More muscular cattle with increased skeletal muscle differentiation will have greater yields and quantity of meat. And now, why was a CRISPR-Cas9 system used as opposed to other gene editing methods? So, CRISPR-Cas9, it's a bacterial immune system adapted for precise gene editing, utilizing a Cas9 enzyme, which has a guide RNA, which leads it to bind to a specific DNA sequence to make the cuts leading to double-strand breaks and mutations and complete gene knockout. Researchers in this study specifically designed a single-stranded guide RNA targeting exon 2 of myostatin. As you can see in figure 1, you can kind of see here just kind of how myostatin works. So myostatin, wild type, is active. It leads to the production of this protein P21, which is an inhibitor of cyclin-dependent kinase 2, which kind of inhibits the proliferation of the myoblasts, leading to minimal skeletal muscle differentiation. Now, when myostatin is not present or active, you don't have that production of P21, you don't have that cyclin-dependent kinase 2 inhibiting the proliferation and differentiation, and you get a lot more skeletal muscle differentiation, hypertrophy. And that leads to what's known as a double muscling phenotype. Now, the methods utilized in this study, the sgRNA targeting exon 2 of myostatin was designed using specifically ChopChop software. And they picked exon 2 specifically after kind of looking at cows that have this mutation present naturally, such as the Piedmontese and the Belgian blue cow, and kind of figured targeting exon 2 specifically would lead to similar results. Now, the Cas9 mRNA and the strand guide RNA were transfected into bovine fibroblasts. After 3 days of transfection, the genomic DNA was extracted, an assay was performed to determine if the myostatin was mutated. Then in vitro fertilization was used to create zygotes. And then different concentrations were used of the Cas9 mRNA and sgRNA to produce the different experimental groups. Then they were micro-injected into zygotes, and micro-injection was used in particular because the researchers spoke about the need for limiting exogenous DNA uptake, and the reason they wanted to limit this is because of regulatory framework when it comes to gene editing, where like an animal that's considered transgenic may fall under different regulations than one that's simply gene edited. So it was more beneficial for them to have less exogenous DNA uptake, which is why they used microinjection, as they believed that would lead to less risk of that, although the risk was still mentioned as being possible. Now, following step 3, step 4, the embryos were cultured in vitro to the blastocyst stage. A portion of those blastocysts were genotyped for myostatin mutation, and then selected blastocysts were transferred into the surrogate cow. Live calves were then genotyped for the mutation type, checking for mosaicism and the editing success rate, and then the calves were then observed as they grew for the double muscling phenotype. Now for the results, as we look over here at figure 2, it has quite a bit of smaller figures too, just looking at 2A, you can see that calf number 17 had the highest success of mutation rate, with 99.9% of the cells noted to have uptaken the mutation, meaning there was no mosaicism, meaning it had the double muscling phenotype. Whereas as you go further back, for example, calf 14, only had 45% mutation rate. Calf 6, only 10.5%. And that's due to mosaicism, where certain cells may still have the wild type versus the mutated myostatin. However, they were still able to achieve a near 100% success in at least one of the calves, showing a proof of concept. They performed a T7E1 assay on calf 17, comparing it to the wild type, a negative control and a positive control, to both determine the exogenous genes, if there was any off-target effects, which they determined none. And then over here, you have a comparison of the expression of myostatin mRNA. As you can see in wild type, as one would expect, it's high. As one would, in calf 14, as we mentioned, had a 45% mutation rate. As you can see here, it's higher than calf 17, which had a near 100%, but still less than the wild type. So it did have diminished myostatin mRNA present. Now moving on to 2E, as you can see, this is Sanger sequencing that was performed to kind of determine what was cut and if it matched. So you have a 12 base pair deletion, which was observed in calf 17, which is why it had such a strong phenotype of double muscling. And you can see over here, at 1 month, the cow number 17, and then at 4 months in B, and you can see the increased muscle. Now coming down to figure 3A, over here you have kind of the target spacer sequence in exon 2, as well as the PAM site that was targeted, and then in B you have various different mutation patterns that were observed. You had some that had a 1 base pair addition, some that yielded a 12 base pair deletion, some that yielded a 1 base pair deletion, with obviously the 12 base pair deletion having the greatest success observed in calf 17. Over here in C, you have the different experimental groups in the blastocyst stage showing the differentiation. And then in D, you have the blastocyst differentiation percentage. As you see, wild type blastocysts weren't really affected by the experimental conditions, differentiating as normal, whereas as you increase the concentrations in the experimental groups of the Cas9 mRNA, you saw a decrease. And in mutation, an interesting pattern that was observed is that the 2X group actually had the highest mutation rate versus the more concentrated 4X or less concentrated 1X group. And just some key implications of this. Of course, 17 healthy calves were produced. One showed a near complete myostatin knockout and double muscle phenotype. No significant off-target effects were noted by the authors. All mutated cows were healthy. They performed some blood analysis showing no significant health issues noted in the mutated cows. And of course, CRISPR-Cas9 did prove effective for improving cattle traits, although the efficiency could be improved for actual use. The purpose of this study, or mentioned early on by the authors, is it's a proof of concept. Editing efficiency now was 81% in embryos, but only 17.6% in alive calves. Authors mentioned in the future, screening the embryos before implantation could improve the success rate. And then looking at the graphical abstract, the problem, again, that the authors were attempting to solve is increased meat demand as global population increases the need for a greater quantity and quality of meat. So the objective was to prove the concept of using CRISPR-Cas9 to induce a beneficial mutation in the myostatin gene in Korean beef cows to cause a double muscling phenotype, leading to more steak. Successful myostatin mutation of a cow occurred alongside a double muscling phenotype, proving the method could work. Thank you for listening. Hopefully you were able to learn something about this. And yeah, that's the story of the buff cow.